Welcome, Pat. I know it's a long way from the Dakotas to Singapore. I assume the weather is different between the two places. Tell me a little bit how you moved from Dakota to Singapore.
Well, that was a long path, Ranga. Started when I was young, of course. I grew up on a small farm in South Dakota and upper Midwest in the U.S. Always was interested in biology, I think because I was a farm boy. So my first love was in biology. Went to college in South Dakota, but knew by the time I finished college that I wanted to move on into a research career. It took a while. I actually spent a few years in industry as a chemist. I worked in Minneapolis in the food industry as an analytical chemist for 3 years while I was trying to decide exactly what I wanted to do next. But it moved on then to do a PhD in biochemistry. I'd become quite interested, particularly in enzymology and how enzymes work when I was an undergraduate. Driven very much by a mentor. One of my professors that I did undergraduate research with kind of inspired me in terms of getting excited about how enzymes worked and how proteins worked, and decided that would be the career path that I would choose to move on.
How did you get interested in biology on a farm?
Well, I mean, it was mainly because you grow up around animals and plants.
Okay.
And I spend all my time working with animals and plants. I just love the outdoors. So I think that I probably was more in love with outdoors biology than laboratory-based science. But I'd have to say all the way back since I was probably 10 or 12 years old, I was reading biographies at that time of scientists, and I was thinking, wouldn't it be just amazing to have a life like that and a career like that? Didn't have to.
Was it a teacher in school or somebody who inspired you, or is it just being
A combination of all of the above. I had a really excellent biology teacher when I was in high school, and a chemistry teacher both. You know, Jerry Opbrook and Roy Risky. I still remember them, and it was just seemed a natural progression for me at the time.
I didn't know you were an analytical chemist.
Yes, that's what I started as. Yeah, so I have degrees in both biology and chemistry from my undergraduate days, and of course the chemistry degree you could actually get a job with if you wanted to spend a few years working. So I was working in a laboratory analyzing vitamin levels in food products.
Well, that should have been, was it interesting? Or?
I thought I had a lot of fun,
Okay.
but after a couple of years, you know, I knew I wanted to move on to something more, and I really felt that academic research would be something that I would like to do, and I wanted to have a chance at it.
So you went from South Dakota to Minnesota?
To Minnesota for 3 years,
And then
I moved to Boston. Yeah,
Boston.
So that was a big change. Yeah, and that was a very big change.
Midwest to the
East Coast, and it was quite an exciting time. It was Brandeis was a small biochemistry department, very committed faculty. It only had 10 faculty. They took about 8 PhD students a year, and we were just immersed in science for 5 years. That was back in the days when stipends were quite low, so you lived in the lab, and I just loved every minute of it, and I just learned so much. And
what were you working on in Brandeis?
at that point, I was working on purine metabolism. I was interested in the biosynthesis of ATP, actually. And there's a particular enzyme in the process called the adenylosuccinate lyase that my mentor had discovered many years before and had linked into a specific step in the maturation of the purine to give you AMP and ATP at the end. It had also been linked, the enzyme had been linked as a potential target in cancer. So that was my first time when I started thinking about cancer biology and potential translational implications of work that I did. So I found out how the enzyme functioned. And also, because of my chemistry background, I was able to develop a small molecule inhibitor and expand upon one that had already been developed. So did my first work with cancer cells in culture, spent a year at Harvard in the School of Public Health with somebody who took me under their wing, Armin Tajan, who was in the School of Public Health there, and taught me how to do tissue culture because biochemists at that time didn't know how to grow cells.
Is it unusual for a School of Public Health to have biochemistry and...
Well, I'd say the Harvard School of Public Health was very different. It grew up as a molecular biology institute as much as a public health institute, and my mentor there, Armin Tajan, who was the chair of the department, and my advisor at Brandeis, Bob Abels, were very good friends. So it was a natural fit. And it was a very short hop on the MRT equivalent, the Green Line in Boston, to be able to go back and forth between the institutions. So got me started in an early stage about how important and how productive collaborative involvement could be. That, you know, if you have a technique that, or you have a question that can't be addressed with the techniques that you have available in your laboratory or around you, you go out and seek someone, and you can do grow your science in ways that wouldn't have been possible if you just stayed in one environment.
And then you went south?
Then I went south. I went to work with Al Gilman, who at that time was in pharmacology, or still is at UT Southwestern. He's retired, but he had come to Brandeis when I was about a year from finishing my PhD, and gave a seminar on G proteins and signaling back when the days where the field was just starting to take off, and I got very excited about it. So I wrote to him, and he offered me a postdoctoral position in his lab. So then I went from Boston to Dallas, which was another bit of a transition, and on to postdoc life, which was, of course, it's a major aspect of one's training along the way to developing as an independent scientist. And it was a terrific environment.
But G proteins are for a lot of things. How did you get into cancer?
Well, again, I wasn't so much in cancer when I first started working on G proteins. I was asking a very fundamental question about G proteins, trying to understand how they moved from the cytoplasm to the membrane, because we knew they had to sit at the membrane in order to function. And as a component of that work, that we discovered that there was a lipid modification that was important in the proteins being able to move to membranes. And then we found that the lipid modification was on an oncogenic G protein, one that's been very widely studied called RAS. And there was a lot of interest in RAS at that time. It was the first human oncogene that was identified. Lots of effort around trying to understand how it worked and how to block its activity in tumor cells. And we found that this lipid modification was important for its function. It was really a side project of something I was doing, but then that triggered me to really move back into cancer biology to understand the role of these lipid modifications in G proteins in general, and how important that was in their functions and in their ability to stimulate cell growth.
So in other words, RAS was your entry into cancer biology?
RAS was my big entry into cancer biology, yes. The G protein that I was most studying in Al Gilman's lab, which was my primary project, is called GZ, or GZ in the rest of the world, and it's found predominantly in the nervous system. So for a while there, people thought I was becoming a neuroscientist, because I was trying to understand how this G protein functioned in neuroendocrine signaling. And in fact, you know, we've just figured it out recently after 20 years. It's always been a side project of mine in my laboratory at Duke. But then my primary interest moved into cancer biology because of the findings with RAS, and because it was a new aspect of control in biology, this lipid modification. So again, we were there at the very beginning of the field. So the fruit was low-hanging.
And there were many, many ways you could have gone, and RAS opened you in a particular way.
RAS opened me in a particular way. Again, I didn't focus completely on it. Mostly what we've been trying to understand over the last 25 years since I've been in the business is the fundamental aspects of how the lipid modification is put on proteins, and what the functional consequences are to the protein that the lipid modifies. And that's led us into control of tumorigenesis. Recently, my wife, Mei Wang, who's a clinician scientist here at Duke-NUS, has moved the field in a different direction. She's found that it's important in controlling aspects of cell death called autophagy, in terms of cells' response to starvation. Also links into cancer and metabolism. So the field keeps expanding, and fortunately we continue to be one of the groups that keep it expanding.
So what made you go to Duke?
Well, Duke, of course, when you're a postdoc and you're finishing your stint, all I was hoping for was that I wanted to establish an independent laboratory at a major research university. And I was hoping someone would give me the opportunity. And fortunately, I had several interviews. In fact, at the time, my first interview in the place I thought I was going to go was Stanford, in a new department of molecular physiology that was created there. But then shortly after that, I was invited to interview at Duke. And Bob Bell, at that time, who was a biochemist and a very well-known lipid biochemist, was starting a new department of cancer biology. And again, this was at the time when I was really starting to think my program was moving in the area of cancer biology. So I visited Duke, and I got very excited about this new department they were creating. And they offered me the opportunity to be a part of it as the founding faculty. And I never looked back after that. I went to Duke in 1990.
So this was before they fused pharmacology and cancer biology
Yes,
together.
yes. So they started a Department of Cancer Biology, and then after 7 years in 1997, that department was merged with pharmacology to create a single larger department. But it started out as its own small unit. There were just 4 of us, actually, 4 new junior faculty that started. One of whom was actually associated with Duke-NUS. Mariano Garcia Blanco and I were hired at the same time in 1990.
Shirish came later.
Shirish came 1 year later.
1 year later.
At that time, Tony Means was recruited as the new chair of pharmacology, and then Tony, one of the first people he recruited was Shirish. So Shirish became my colleague in a separate department, but he was already at that point in time rapidly became a friend, colleague, and a mentor. In fact, he helped me get my first grant.
Okay.
Yeah, because he's a whiz at helping people tune their grants to make them understandable, particularly to reviewers.
So what was interesting about your time at Duke?
I just thought Duke was a terrific place. I always, I love Duke. I'm just incredibly happy that I'm still a part of Duke, mostly in Duke, Singapore, but I still spend time in Durham every year. It was just a great collection of scientists. I thought that the esprit de corps of the faculty were very interactive. They were very committed to training. They were committed to excellence. There was a lot of mobility of the faculty. They were very collaborative. It was just a great environment to be in, and I think it still is today. Yeah.
So when Singapore came on the map, how did they get you to move? You were the first person.
Yeah, that
without
story.
you, the school would have probably not started. So,
well, thank you. But and I've been very happy and proud to be a part of the development of this school. I got involved quite early in 2003. The vice chancellor at Duke, Bob Tabor, who was in charge of kind of Duke's international ventures and its forays out into the world, was the architect of the plan to develop Duke-NUS, and he was the primary point of contact in the negotiations with Singapore when we found out and they approached Duke about establishing this new medical school. And for some reason, Bob Tabor thought that I would be potentially interested in it and might be able to contribute at least in the planning stages. So that's how you always get pulled into these things. So I was invited to join the planning committee in 2003 to join a group that would come over to Singapore and begin discussions around what this school would look like. If Duke was really going to do this, what did we want the school to look like, and how did we feel that we could really contribute to the development of the school, and in return, what did we think that, how did we feel that this would help Duke grow as an institution? So I made my first trip with a contingent of Duke faculty. There were, I think, 5 of us that came over in November of 2003, and by the time I finished that trip, I was pretty hooked on it. And
okay.
Amazingly, my wife was also interested in the endeavor as well.
So what hooked you when you came?
I would say it seemed like there was a real commitment to excellence and the development of biomedical sciences here. We saw opportunities to be in on the start of something that we thought was going to be very special, and that something that we could really contribute to and make an impact in. I was, at that time, I wanted to grow my program. I wanted to grow, I should say, in terms of my development as a professional. I had considered a couple of department chairs. I had looked at a couple of department chairs around the U.S. thinking, well, maybe I would take over a department and help shape a department. But the opportunity to build a medical school from scratch in a country where the commitment was clear and evident on all parties, where it was clear the resources would be available to do something that would be quite special and also we felt could really have an impact and grow research programs in ways that wouldn't be possible at Duke or probably any place else in the U.S.
So how did you get about recruiting people? Because that was your first job.
Yes. Well, the first job was actually to help build the administration so that we could get the school off the ground. So it was about a year after we were here. So we came in August of 2005, and we really launched recruiting efforts in the fall of 2006. Initially through a series of contacts, posting descriptions and advertisements in places like Science and Nature, and spreading the word about what we were doing here. I spent a fair amount of time visiting institutions and making sure I was connected with colleagues across the U.S. and Europe and around the world about what Duke was doing here. And Sandy Williams was very much engaged as well. He's our founding dean. Again, all the Duke people were now letting the world know what we were doing. And basically we just put the word out that we were looking for a cadre of faculty.
So how did you manage to attract people? What did you have to say, or what did you have to say much to attract them?
Well, I mean, you got to make a pitch, of course. And really what draws people here, I think the first people that came, like David Vership, he was excited about the opportunity to build something. He was the first program director that we recruited. Now there was a connection. He hadn't even known that I had come to Singapore, but I had put these notices in Science and Nature that we were recruiting faculty. David and I had served on a study section on a grant review panel for the American Cancer Society for many years, so I'd known him for years. And he saw this with my name on it, and he sent me an email and he said, "What are you doing in Singapore?" So it was clear from the email that he was a little bit interested in what we were doing in Singapore. So I called him that next day, and we had a good conversation, and it turned out he was thinking about doing something different as well. He'd been in Utah for a long time and was looking for an opportunity to build a program. I was looking for someone that would come out and help build our cancer biology program. So he came out and visited, and you know, again, like everyone who comes here, I think the place is captivating on a number of fronts. Just the environment, the people. I mean, it's an easy place to live. The people are all very, genuinely enthusiastic and committed. It was clear the resources were going to be available to build a special type of program. And so we saw opportunities on many fronts. I mean, I would say that everybody who comes here, they come for different reasons, but they all see opportunity.
So when the planning was coming along, how were the decisions made to have 4 or 5 research programs?
Well, initially that came from discussions with Sandy Williams and then Chancellor Ralph Snyderman, but about halfway through the planning process was when Ralph retired and Victor Zhao came on board and immediately embraced the idea of this new medical school as well. So we wanted to build programs that were very translational in nature. We didn't want to do something along a traditional line of departments of biochemistry and pharmacology, et cetera, but we wanted to come to really build programs that we thought could take fundamental discoveries and move them rapidly into a clinical application. That's what Duke became very keen on, the potential of starting a second medical school, because they could test out models, both in terms of how science is organized and how research programs are organized and connected in really substantive ways into the clinical enterprise and into development and application of technologies, but also in terms of having access to patient populations and a health system that would allow one to test out new ideas in healthcare development and delivery and design of health systems and delivering healthcare to communities. So we started initially with 4 programs. All 3 of them were laboratory-based, and we felt that they would connect substantively to programs at Duke, as well as there were important medical needs that were unique to Asia, or there were unique opportunities in studying these diseases in Asia. Cancer, around neuroscience, around cardiovascular and metabolic diseases. And then the health services research program was to test out these ideas and potentially, new ways to deliver healthcare services and really make a more immediate impact. All 4 were areas that Duke was trying to develop and was developing, and saw opportunity to expand that development in a unique environment. Infectious diseases was added shortly after we came over, primarily at the invitation of the government, because of what they had just gone through with SARS and the concern about new emerging diseases. They saw that what we were developing on the other front, so they asked us if we would, in fact, add a 5th program around emerging infectious diseases, which was quickly embraced, I would say, on the Duke side. Again, feeling that we had unique opportunities to do that type of research in Southeast Asia.
So you've recruited a great group of faculty.
Yeah, so they've been a terrific group of faculty.
Postdocs and others. Was there any challenges that you felt?
There are always challenges. I mean, but challenges are what keep the job exciting.
Mm-hmm.
I mean, it's always, and the challenges come from many aspects. One is we're always on the lookout to try to identify really talented Singaporeans that have left Singapore for one reason or another to pursue their careers. They always make great recruits because, again, the opportunity opportunities that have been created because of the Biomedical Sciences Initiative in Singapore. So on one side, we're trying to entice Singaporeans to come back to Singapore. We're trying to bring in and expand talent from across the globe. Again, it's a very multinational society here, and in particularly bringing in people with very diverse backgrounds from very diverse types of institutions and bringing them together in a single place where they can interact, I think really helps spur new innovation and new ideas around research programs. So each one, I mean, every recruit's a little bit different, but always, again, you're trying to make a connection where you see what's the opportunities that we can provide that you might not have elsewhere, and what's the environment that we can provide that will help your science grow and flourish.
So looking back from South Dakota to Singapore, are there lessons learned that could be helpful for postdoc students who come through Duke and come here?
Well, I would say the lessons learned are you'll always do the best in something that you're passionate about. You always you want to follow the path that gives you the most excitement, because that's going to be the most fulfilling as your career develops. Some people are very smart and they can do very well, but they don't rise to the same level if they don't have a passion and a genuine excitement about what they're doing. So we're always looking for people that are genuinely excited and passionate about what they're doing, in whatever aspect and whatever they're undertaking, because they're the people that really can push things forward. Not only do you feel that you're growing in your career development and your professional development, when you have people, when you're excited, people around you are excited. And that helps you attract the very best trainees, the postdocs in, and really inspires a culture of excitement and excellence about what one does. Yeah.
So passion, curiosity?
Curiosity and passion, I would say, are the two strongest traits. It's, because again, everybody who gets this far in life has already the intelligence. Those filters have come in earlier in life. So when you get people that have already finished their first degree, and particularly that they're been admitted into a PhD program, they're all smart enough to do good stuff. They have to be curious, they have to really want to make a difference, and they have to be excited about what it is they're trying to find out. Yeah.
Now that the school has been up and running for several years, what excites you going forward?
What excites me going forward? That's a very good question. It's because we're reaching almost a steady state in the growth of the Duke-NUS as a single institution. What excites me now is being able to really take things to the next level and connect in a broad way across Singapore and internationally as well, to ensure that our programs really do have the impact that they were designed and supported to do. So we have many challenges ahead, as we all know, in terms of being able to really substantively integrate our research programs with the broader biomedical sciences and the entire health system of Singapore, so that we can have that impact that we all dream of and that we all came here to be a part of. So what excites me is the continued growth and the opportunities that are going to come from these collaborations and partnerships that we're forming with institutions and across Singapore, particularly in the Sing Health system, of course, with our academic medicine initiative, but in all other aspects of interactions with our partners as well.
Your science has gone very well over the years. Where do you think it's heading?
I'd like to see the science now. Spent 25 years really trying to understand fundamental aspects of G protein signaling and these lipid modifications. We have clear evidence now that if we can go in and manipulate these processes in the right way, we can impact the growth of tumors, including studies in mouse models. So what I'd like to do over the next couple of years, in collaboration with my wife, who's driving the kind of clinical sciences development, is to move this into clinical application. We'd like to be able to test the models that we've developed and the hypotheses that we've developed as a result of our science in humans, to see whether we can, in fact, impact on tumorigenesis and have an impact on the cancer problem.
So passion, curiosity, and then translating it to benefit people.
Yes. Yeah. At the end of the day, if I retired some many years hence, There were aspects of my research program that were in clinical application, I would retire very happily. But like I said, that's quite a long ways off yet. And I very much want to continue to be a part of it as long as I'm capable of being able to be a part of it.
Any final words for people here at the school? Anything they should know about you that they don't know about you?
No, I mean, I think I know people here quite well. I mean, it's been a terrific group. We have just the faculty, the staff, all the researchers here. I think they're a very inspiring group. I've just been, like I said, I've been quite fortunate and proud to be a part of the development of the school. And I look forward to being a part of it for many years to come. I was talking the other day with some friends back when I was at Duke. I was back in the U.S. just last week, and I said, I'm looking forward to the 25th anniversary of Duke-NUS coming back, you know, and seeing really what this school is then, and reminiscing about how we all got it up and going.
Thank you, Pat.
All right. Thank you, Dean.